Hello and welcome to 3 things everyone needs to know about PFAS. If you haven't seen part 1 and part 2, that's okay. This is part 3. You can go back and watch part 1 and part 2. You don't need to see the first 2 to see this one. Today, we're talking about structural diversity of PFAS compounds. So I used a quote here, quantity has a quality all of its own. This is maybe attributed to the Soviet Union in World War II, maybe Joseph Stalin. It's a little unclear. It might just be one of those quotes that's seen everywhere. Not too sure. Anyway, it applies really well to PFAS because that is why I have chosen diversity over toxicity for this third topic as being one of the most important aspects of PFAS, because you have so many different types of PFAS, so many different structures, it may be impossible to even know the toxicity of all of them in the first place. That's the paradox here. I use the word paradox a lot today, I think. So first of all, PCBs. I always like to go back a little bit in history. PCBs used from 1890 to 1970s and 1980s until they were phased out. Polychlorinated biphenyls, you have up to 5 chlorine on each of those biphenyl rings. And what you get is, in addition to a very persistent, a very bioaccumulative, and a very toxic compound, you get actually 209 different structures just based on how many potential ways you can move around those chlorines and how many combinations. So it makes it a little complicated when they were first researching PCBs, because some are not very toxic at all, or some are very toxic just based on how, what structure and those minute structural differences. And these are some capacitors from around 1980s. The left one, I think, is from Russia, Soviet Union. The right is from Germany. So you think about those 209 different potentials and how just the arrangement of some chlorine atoms is going to make a big difference. Let's apply that to PFAS. So if PCBs are complicated because there's 209, PFAS is on a different scale. It's not even on the same scale, because this basic structure of PFAS, meaning that at least one carbon is fully fluorinated, as long as these 2 R groups aren't hydrogen, this would be considered a PFAS. It's an alkyl, fully fluorinated carbon there. So those R groups can be anything. It can be, 100 chain long carbons to make kind of a polymer long thing, or it could just be some reactive groups like sulfonamides. It can be anything. There's no definition here. That's what gives PFAS their diversity, is that it's a wide open class. It's just something that contains a perfluorinated carbon. When you Google how many PFAS are there, because it might be a question a lot of people have, you get different numbers, but usually the range now, the consensus is there's 4,000 or 5,000 that have CAS numbers, or have been reported in the literature or in patents, or have been produced in some way, or just theorized. That's about the number of potentials out there, and this is good work. I think it makes sense, but it's definitely not everything. There's so many more potentials. For a lot of these, you could extrapolate multiple chain lengths. If you add an extra chain length that they didn't think about, just an extra CF2 to all of these, you double it right away, right? So you instantly come with 10,000. If you replace each of these with 1 extra chlorine instead of F, you're going to double it. I think that shows the potential of the diversity here. Again, our structure, those R groups can go, any potential. And so what I tried to do here is I tried to draw with a little counter to count how many different structures I'm drawing here. So I tried to draw I'm kind of like randomly drawing, adding some oxygen, some sulfur. Some of these are real PFAS, by the way. Some are just imaginary makeup PFAS, but a lot of what I'm drawing here is kind of based on what we've seen out there. But the counter's going up because each time I'm adding an atom or taking one away, we've got an extra potential compound, right? So you can see how fast this is getting, to where if you did this for a few minutes, we would quickly overtake that 209 for PCBs. So just in what I'm doing here, adding carbons and sulfurs and just kind of having fun, you get a number 51. And so 51, that's a manageable number, but you have to think about the potentials with PFAS. 51 times, and like I mentioned a second ago, take one of those fluorines and replace with chlorine. Okay, that's a common thing that's happening these days. Boom, we've doubled. So for every one of those structures I draw, you could draw one with one substituted chlorine. So now we've doubled. Maybe one of the F's is substituted with a hydrogen, so that's doubled. And actually this should be factorial because it could be one hydrogen and one fluorine or chlorine that's replaced, right? But I'm just doing multiplication. If there's an extra ether, like shown in this structure, between a CF2 group, and then the chain length of those CF2s, that could be from anywhere from 1 to 18 to 25 to 50. I just assumed it was up to 18 chain lengths. Now our 51 has just been multiplied to 7,344 potential structures, just in what I drew. That doesn't mean that they're all organically feasible to be synthesized, but it just, again, reiterating how diverse this class is and how quickly you can get to very big, overwhelming numbers. The question of today, PFAS diversity, how do you solve a problem that you don't know exists? So with persistence and bioaccumulation, we've had a handle on that, on the PFAS we know. But what about on all these we don't know? What about all of these that are being synthesized and nobody knows about them? We don't know about their persistence. We don't know about the bioaccumulation. So in some aspects, this diversity brings more risk and needs more care because it's just going to multiply that persistence and bioaccumulative, right? Because it adds that unknown factor. So I found when I was thinking about it, trying to organize this, I found 4 main areas that diversity really impacts PFAS risk assessment. The first would be environmental modeling. Modeling is extremely important in the field of environmental risk assessment. Places like the EPA, they use models to do what they wouldn't have time to do in a lab. To do a lab study on a compound would take years. To run a model, it can be very quick. It can be almost instant depending on what the model is, and can give you an idea, good or bad, risky or not risky. Is this red, yellow, green on our persistence scale? The problem with PFAS and their diversity, the potential is unlimited. It's not like we can make a model that says, okay, if there's chlorine here, what's it going to affect? We have to take into account structures that we don't know what the structures are, and that's impossible. So when you try to do this modeling for the risk of PFAS as a class, again, it's impossible because we don't know what they all are. That's a paradox, right? So as the structural complexity goes up, our model effectiveness goes down, and our whole ability to model in general goes down. We don't know what to model for if we don't know what's there, right? Okay, detection. This is my field. This is my current job is working in PFAS detection. And with the detection of PFAS, not only do we need to be able to do the work where you go out and say, what is contaminating this site, what is in our bodies, you need to be able to say how much. Saying what is great because it's going to push the field forward, but without being able to quantitatively determine are levels going up or going down, like Paracelsus said, the dose makes the poison, right? So at a very low level, maybe they're okay, maybe not. We don't know that till we've tested. And currently, in the last 20 years since PFAS really blew up in 2000 and the interest really, really got kicked off, we've gone from maybe 2 standards, PFAS and PFOA, to maybe 90, 100, 150. I just plotted here, these are the names from Wellington's website that I found that are unique compounds that aren't kind of branched isomers. I found 91 from Wellington. I'm assuming there's some others out there, so I'm calling it 150 potential analytical standards compared to what was our number, 5,000, 10,000, a million potential PFAS. This is a huge limitation. It's a huge, it's what lends a lot of the risk to PFAS as a class. Toxicity, which I will talk about, I promise, in another video, but it's when you're talking about toxicity and PFAS, we are able to look on a compound to compound basis, what's toxic, what's not. We're able to do some modeling on this, but again, till we know what structures are out there and what the potential structures could be, there's no way to know if it's toxic or not. We have to identify what's out there first before we can determine the risk. And since identification, which we just said, is nearly impossible because of the number of compounds, then where are we? We're stuck. We're just stuck with a toxicity detection loop here. So that's why I think the diversity almost trumps its toxicity, because there's that unknown factor that scales it up to infinity for potential for toxicity. And last is regulation. So with a huge diverse class of compounds with a huge diverse potential for different structures, how do we regulate it? It's daunting. We have an industrial application by this, represented by this bronze or brass faucet, which probably contains lead if it's brass. We have this faucet of source of PFAS going into the environment, getting to the consumer. But we have a role to fill. We have an industrial application, and that's fulfilled by a PFAS here, right? And what happens when we regulate that is that an application still needs to be fulfilled by something. And PFAS are unique special butterflies. It's hard to find a replacement that's not also a PFAS. So it gets replaced with a different PFAS. And when new regulation comes in, it gets replaced with a different compound. Like this will go on. We're at number 3. This could go on for 5,000. And this is the problem with regulating single compounds on a compound specific basis, is we still have an application that needs an alternative, and that alternative is going to be PFAS. So regulation of an entire class of PFAS then is complicated. Compound by compound, I just showed that logically it's not effective. Doing an entire class is also flawed. How are you going to choose a class? Are you going to say, is it a perfluorinated acid that we're regulating? Because what if a perfluorinated acid is required by something and that's the only way to get it done? Or what if a perfluorinated acid excludes some potential safe alternative perfluorinated acids that we could have used as replacements? It's hard to know without knowing everything that's out there. So how do you regulate PFAS? It's the trickiest question to answer, I think, in environmental regulation right now. Maybe not the trickiest. I'll go ahead and call it the trickiest. Okay, so scary stuff today. I didn't realize part 3 would be the scary talk of these. With persistence and bioaccumulation, they're pretty scary, but I think it's because of the unknown factor of PFAS. The diversity leads to unknown. Unknown is scary because potentials are unlimited. So to demonstrate this, I did some art. My name is Simon and I like to make drawings. The art here, if you look at it, I'm calling it a PFASberg, because it's showing that the PFAS we know kind of up at the top are really the tip of the iceberg. We don't know what else is out there. This is an artistic representation, not to scale. The scale would actually be a very tiny part of the PFAS iceberg, the PFASberg above the water there. So with the PFASberg, tip of the iceberg, that concludes today. Structural diversity of PFAS compounds. The potentials, again, I said it 100 times, are unlimited, and trying to assess the risk of PFAS puts you in these circular loops. How do you know the risk if you don't know what's out there? How do you go find what's out there when the potentials are unlimited? So that's it for today. Please subscribe. Please give a thumbs up if you liked it, and tune in for the next video, which probably is going to be toxicity. Could be maybe the different regulated methods out there, drinking water. Could also be human exposure. I think it'll be human exposure. Anyway, take care and bye.